Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, here we are again from our break. From where we stopped, remember, we were talking about what we got from the light-dependent reactions to be used in the light-independent reactions, the one we normally refer to as Calvin cycle. A great scientist that discovered the stages, the shortcuts, the routes, the channels, the corners, and the curves taken by a carbon dioxide molecule to move through a whole plant, leaf, chloroplast, until we get glucose out of it. But the question is, how did he track this? The answer to this is going to be, we shall look at the experimental design of what he did to be able to track. From here to here, after 1 minute, carbon dioxide has moved to this phase. Now it has changed to this format. Now it is like this until finally glucose forms. In the same way when you're cooking at home, you keep checking the food, is the salt ready, how far has the food gone, until finally the food gets cooked. So you're able to track, after 5 minutes, what is rice like? After 20 minutes, what is rice like? That's exactly what he did. He tracked the molecule of carbon dioxide from minute to minute until finally glucose was able to be produced. So he could know the changes. Maybe the enzymes involved as well. And also, when we talk of intermediate products, halfway, something like that. Remember that we are saying that we are in the light-independent reactions taking place within the stroma, another part of the chloroplast. We are from here. I don't want you to think about it anymore, but think about what it has given us, these two. So, when we talk of fixation, get the carbon dioxide, add it to another molecule, form another compound. What exactly do we mean? So here we go. The process of formation of glucose molecule has stages, and the first one is the fixation of carbon dioxide. We have a lot of compounds involved. You have ribulose bisphosphate. A ribulose is a 5-carbon compound, as you can check the circles indicated here. Each represents a carbon atom, so this is a 5-carbon, right? This is carbon dioxide, 1 carbon atom. The reaction occurs in the stroma, the other part of the chloroplast without the pigment, the other part of the chloroplast where we have a pile of enzymes. So it's the matrix, a concentration of liquids and solutes and enzymes, all conditions to favor the process of fixation. So the ribulose bisphosphate is an acceptor molecule. Now guess what happens? The moment these become 6 carbon atoms, the compound formed is so highly unstable that it breaks into 2, forming a glycerate. Sometimes you refer to as PGA or phosphoglyceric acid. 2 of them. Ribisco or ribulose bisphosphate carbohydrase enzyme catalyzes the reaction. Like we just talked about, the splitting is the high instability of the compound once the 5-carbon atom adds here. To form 6, highly unstable, break into 2, 3C compounds. So this is a 3-carbon. This is a 3-carbon. Maybe I need to write it. So we write 3C, and we also have another 3C. That's the fixation. That is the acceptance. But what exactly happens here? Once these have been produced, they are unstable up here, produced as 6, which breaks down to give 2 3C compounds, now stable. We have a problem now. We received carbon dioxide. Check this very well. We became 6. And then we broke down into 2 parts as 3,3. Stable. How shall we move from here? Until it rains, or what? Now we need to get back to our stores from the light reactions. The substances that were produced during the time when light was striking the pigments are now of great importance. So what happens? That the stable 3C compound is going to use our ATP to get activated. So we are going to consume ATP here. As you can check the arrow from here to here means we have consumed ATP and we now have ADP. Once we have activated this, then we are in position to reduce. There is no reaction that occurs before a compound has been activated. So it's active, ready for reaction, ready to undergo change. And which change do we have here? To reduce it. So interesting. We added a carbon atom, they became 6, it got unstable, broke down, stabilized. We need to activate it again, make it angry, then finally force it to pick up a hydrogen atom. So what do we have here? ADP, if we are to have this reaction continuing, this has to go back to the light reactions and pick another phosphate to again have ATP. So NADP H, after forming NADP positive, will go back to the light reaction, the light-dependent reactions, to be able to come out like this and be able to reduce the compound again. So the triose phosphates we do have here are a reduced form. A reduced form. 2 of them, remember. Now, what next do we have? Remember, we are here. That most of the triose phosphates produced when we have used NADP H. I want you to look at this very carefully. Please go with me. The language. Icyongereza. Ntabwo ari Ikinyarwanda. Si iby'iwacu. Most, it means that a percentage that is above 50. It means that a greater portion of what we have here, a greater portion of what we have here is used. It is funny that you've moved all the way through, suffered with the light, suffered with the carbon dioxide, got reduced, got activated, and once you have reached here, instead of forming enough foods, 5 out of 6. So when you convert this into percent, you can imagine a greater part of what we have here is actually used to give us our NADP, to give us our acceptor molecule, which we do have here, RUBP. Remember, this is supposed to receive carbon dioxide. And then, once it has received it, it disappears. We don't see it anymore. We can't track it now. It's hidden within the reaction intermediate compounds. But by the time the reaction is over, that is our now task. It is in here, it's in here. Now, how do we get it such that we can have the reaction resume again? Interestingly, we are lucky that a greater part of what is embedded here shall be used to bring our RUBP such that, check that very well, kujirangu. The other lady standing by the gate at the reception can continue to bring guests in. Akamwicaza ku ntebe muyobozi byubahiro byinshi agasubira hanze akafata undi mushyitsi akamwinjiza, like that. That is the role of IBP. So once we have it hidden here, it means that wa protocol wacu aragiye nawe yiyicarira mu bashyitsi, ariyicara ngo nabo bamusariva agafanta. Kibazo nuko yahava afite umuheha gusa. So this lady has to go back. She has a badge to identify her as part of protocol, so she has to go to the gate to receive more guests. That's exactly what is happening here. IBP has to be regenerated, but at a very huge cost. One, that we are going to consume energy to bring it back. I don't know how many times when you go to weddings you've seen, once these people in protocol get tired, kavana muzamwe tondende akambara masai akagira hakicara noneho na ya badge akayikuramo kugira ngo bataza kumenya hanari aba yarushe. So in the same way, trials phosphate, a greater part of it is used to bring back RUBP to be able to continue receiving carbon dioxide, because it is a cyclic process. Fixation of carbon dioxide is cyclic, it's continuous, but at a very huge cost. Much of what we have produced after receiving it is used to bring back the acceptor. So it's very important. So when you have weddings and any other functions, please give huge value to these receptionists. They do so much. They move a lot of miles and make sure that everyone sits. So you need to give them respect, and they have to eat even before others. So here we go. It will require us a lot of energy to bring it back. The reason as to why. Light reactions have to continue so they are able to produce this ATP. Otherwise, the reaction would finally reach here and stop, since we cannot bring back the receptionist, the protocol lady, the protocol guy. He's going to sit among the guests and that's it. So he'll keep locked here. And then more guests will stop coming in. More carbon dioxide will stop coming in because there is no receiver for it. Now, the 1 out of 6, the remaining component, sum of that. I love this. I don't know whether you really don't see it. This is 1 out of 6, sum, a little of it. That's what it actually means. Most rejuvenate, bring back RUBP. Some produce the glucose. That's unfair. I would want to think so. So we have our glucose phosphate that will undergo a lot of changes until we finally can have starch by condensation, biological molecules, senior 4, you remember that? Add glucose plus another glucose, then you get maltose, continue adding others and stuff like that. But the question also we need to think about is, once this glucose has been produced, does this arrow tell us that actually glucose is a product of light-independent reactions? The Calvin cycle, now check this, very common in assessment. Its products for it to restart again are here. I want you to pay attention to this. The triose phosphate is the main product of the Calvin cycle. Because we had this before, so we can say it's a product. And for us to have glucose, those are other proceeding reactions which are not part of the cycle. This is just an extension. So when we talk of the main product of the Calvin cycle, come on guys, it's here. So, putting it together, our cycle now. I want you to take a look at it. Bridge all the small pieces we have been looking at one by one, and look at the general overview. Follow the arrows. Follow the path. Check where we look at when we say or check what we look at when we say the product of the cycle is here. It's not this. This is the overall at the end of the day when other reactions have gone or have taken place, as we shall also see. Our focus here has been light-dependent reactions for producing ATP and NADPH, to use them as a backup to understand what their role shall be, how they link with the Calvin cycle. And now here we have our Calvin cycle. Main components: fixation, carboxylation. To carboxylate is to add a carbon atom to a carbon compound. Chemistry, right? Organic chemistry. These are organic compounds. An enzyme is needed, ribosco. Compound produced is so unstable that it quickly breaks down. The stable compounds now will have to consume the ATP from the light reaction. Can you check now how important it is? And then, after being activated, we do get our reducing power taken up to reduce the phosphoglyceric acid or glycerate phosphate to a triose phosphate, PGALD. You come across all those words because I really strongly want to think that even when you're back home, you're having texts with you, the textbooks. It's not just about sitting and watching TV without a pen or anything. This gives us an overview, a general representation of the reaction. So once these have been produced, like we said, so much energy is again needed to bring back the RBP. Our protocol lady is here. Our protocol gentleman is here to pick more carbon dioxide. So the process can continue. It's cyclic. Then part of this, a very small fraction, remember we said 1 out of what? Out of 6, right? And then the 5 out of 6, the bigger chunk, is what is used to produce this back, while a small portion is used to form glucose that can be turned into a variety of forms. So it is from here that we can have lipids here. It is from here that we can have glucose. It is from here that we can have proteins. It therefore means that the same component here on this arrow, it is from here that we can have proteins, amino acids, right? It is from here that we can also stretch another arrow and put there lipids. So check that. These 3 foods, so we have the carbohydrates, we have the proteins, and the lipids. They branch off from a triose. So never should you think that the plant will have to first form glucose, then it will convert glucose into a protein. Oh yeah. So what are we talking about here? This end product of the Calvin cycle is from where we get the other foods manufactured. That's for another lesson. So what are we talking about here? That this glucose, once produced, can be turned into starch by condensation for storage. The glucose phosphate can lose the phosphate ions and turn into fructose, and then added to glucose, and then we turn into sucrose that can be transported. Remember that the transportable form of sugars after manufacture is sucrose, a disaccharide of a fructose and a glucose molecule. In the same way, maybe the glucose produced can also be used in formation of cell walls, like we looked at the very beginning. The functions of the glucose produced: starch for storage, glucose for continuing respiratory needs, and 3, cellulose formation to build the cell wall. Now, here is one for you. Are we in position to explain how? Wonderful command questions. An explanation means you should give reasons as to why, but also how they want the process. Nigute. From which step to which step. When you say how the light-independent reactions of photosynthesis, the ones we just finished, the Calvin cycle, depend on the light-dependent reaction. Now, this takes us back to our objective, the link between stroma and the grana, the link between ATP, NADPH, and carbon dioxide, until we have the glucose. Steak or score, amanota nangahe atandatu. Ni menshi. So make an outline so quickly, and we go through this now. Let me simplify it for you. These are the considerations. If you're to get the 6 marks, based on what the question has asked you, what is the examiner looking for? There. Right? So, we need photoactivation, exciting the electrons, leading to formation of ATP and NADPH. As the electrons move, they are pumped, ATP is produced, photophosphorylation. Phosphorylate is to add phosphate compounds to a compound. We remember that we need so much of this because we need to bring these back every cycle. We need more of them. So the light-independent reactions that fix carbon dioxide to make glycerate 3-phosphate, to be reduced, we need them, but also activate them so they are also reduced, we need ATP. And at the same time, the same energy needed for recycling the reaction. Now, that is how you would respond to that question. I hope you have your camera with you and you've taken the caption, because 2, 3, 1, we are going away, and wrap it up with the whole clip. And there we go. Pay more attention again. We are wrapping it all together. All the things we have looked at, the light reactions, the characteristics of light as a wave, one as a particle, the photon. Of all the solar energy that is released, what is important for us? When we talk of the electromagnetic spectrum, we only need So we continued. The tape is still playing, and we now need to check. That is the wrap of the light reaction. Now we're going to see the dark reaction once this is over, which should be able to take around maybe 2 or 3 minutes extra, and then we should be done. The exciting of the pigments, transferring of the electrons across the electron transport system, and then you have photosystems, and then our pump we're looking at. The piling of the protons. As you can check, every time water splits, we have an addition. Oxygen is released by diffusion if it's not needed in the leaf for respiration, and then more electrons will replace what has been lost by PS2. Handed over to PS1, and then these electrons will be the ones to reduce the deep H. So the H on NADIP is actually to give us a sense of the process of reduction, that a compound is reduced. So do not think that it is H that goes there, it's the electron. We are looking at it in terms of electron addition as a reduction. So when we finally head into what we look at, once we have these products, and we have them transferred to the dark phase, which is no longer used, and we talk about light-independent reactions. When we have the proton pump producing ATP and the deep H within the green section of the chloroplast, the thyroid structures, where shall this finally head into? Exactly there, where we have what we are talking about, where ATP is used, where NADP H is used, where we have the product, and the cycle continues to bring back. So we have fixation, and the breakdown. So follow the path very carefully and check as we wrap this up. So with this, let's end here, and as you continue checking, we should be in position to see how the products are produced. And with that, thank you so much.